I guess it's 1 o'clock, so we can get started. And I'd like to welcome everybody to our seminar, the Single Cell Spatial Analysis Program seminar. And we're very lucky today to have a very big name in the field of spatial analysis joining us. And so I'd like to take a chance now to introduce Dr. Evan Macosko. He's an institute member at the Broad Institute of MIT and Harvard, where he directs a lab in the Stanley Center for Psychiatric Research. He is also an associate professor of psychiatry and neurobiology at Harvard Medical School and attending psychiatrist at Massachusetts General Hospital. He serves as a co-PI for the BICAN Brain Cell Variation Grant, and his lab develops new genomics technologies and deploys them to understand pathogenic mechanisms of brain diseases. So I'm very excited to have Dr. Macosko sharing some of his research and thoughts today with us in our program. Evan, please take it away, and thank you very much.
Thank you, Evan, for that introduction, and thank you all for joining. Let me just share my screen. I assume you are able to see everything nicely now?
We can see, yep.
Great. Yeah, so as Evan mentioned, I'll be sharing with you kind of a combination of efforts to leverage some of the new tools that our lab has developed over the last several years in spatial transcriptomics especially, and then also tell you about a new technology that I think is going to be particularly impactful for kind of uniting the worlds of single cell analysis and spatial genomics analysis. So the first part will be kind of more, a little bit more biologically focused, analysis focused on a large data set we generated, and then the second part will be more technology focused. Our lab is really focused on trying to leverage these technologies to understand the brain, which is arguably sort of the greatest marvel of biology. The fact that there's this tissue that is composed of cells just like any other tissue, but these cells have a lot of very strange, unique properties that have been appreciated really since the beginning of modern neuroscience. And these properties allow cells, of course, they have many complex connectivities as well, but the cells seem to allow the tissue to do many unique things that cannot be done by other organs. And so we're really interested in trying to understand, sort of develop approaches that allow us to sort of systematically understand the brain, and then to leverage that understanding to understand disease states. So the kinds of basic questions we're interested in are, what are the types of brain cells that exist in total in the brain? What are their functions? What are the effects of the disease and can we leverage these different cell types for therapeutic purposes? And I just wanted to give a little sort of overview of why this is a particularly exciting time to be leveraging genomic tools for this particular purpose. Historically, genomics is really kind of focused in a way that was very removed from tissue. Analyses like whole genome association studies or bulk analyses of gene expression using gene microarray technology, those sort of older technologies didn't really give us a deeper view, a sufficiently contextualized view of what's happening in a tissue. And then the invention of next generation sequencing 10 years ago really allowed us to combine that sequencing technology with new approaches in microfluidics and barcoding to now be able to sort of sample gene expression and also epigenetic regulation in individual cells and in spatial context, really moving us more towards what we kind of think of as tissue genomics. Because I think this next era is going to be a lot about taking these measurements, these omic measurements, and combining them with cell biological measurements so that we can really start to understand tissues in a much more holistic way. So our lab tries to tackle this sort of overall challenge in sort of three major ways. We invent new technology. We leverage it to develop comprehensive atlases of cell types in the brain. And then we also try to apply it to understand cell type specific responses to disease. And so first I'll tell you about a recent study that will be out next month, in which we've really systematically characterized all the cell types that are present in the first time in a mammalian brain, in this case, in the adult mouse brain. So really answering this first question that I articulated earlier about how many types of brain cells are there throughout the brain. And sort of related to that question, or secondarily to that question, is how are these cell types distributed across different structures in the brain? How different are they from each other? And what are some features or emergent principles that come out of this cellular diversification that kind of just teach us something about how this complexity is orchestrated and its function in the brain. So to tackle this effort of trying to define all cell types in the brain, we leveraged two major approaches. The first is single nucleus RNA-seq, which is really based on the development when I was a postdoc of drop-seq, which is the first technology that allowed one to systematically barcode RNA using microfluidic droplets. And this is just one of those first t-SNEs we generated from the mouse retina. And what we immediately saw from these 49,000 cells is just the immense complexity that's present in nervous system tissue. And the exciting thing is that this technology, of course, scales very well to the size of a whole brain. And then next, of course, what was missing was the ability to localize these gene expression defined cell types in particular areas of the brain. So partnering with Fei Chen's lab over the last several years, we developed a technology called slide-seq, which basically takes these same barcodes that were found, barcoded beads that are found in drop-seq, and then puts them on a microscope slide so that you can now put tissue down on that microscope slide, capture RNA from individual 10-micron pixels, and read out all the RNAs that are found at that 10-micron location. This is one of the first experiments we did in the hippocampus, where we just clustered these beads as if they were single cells, and we then looked at their positions in spatial coordinates, and we found that we could recapitulate, in this case, the structure of the mouse hippocampus. And because this technology is so scalable, we realized we could do this across an entire brain. So that's what we did. Chuck Vanderberg and Naeem Nadaf dissected the mouse brain into 100 individual dissections, and then ran 6 million single nucleus RNA-seq profiles on those individual dissectates to generate this t-SNE, which I think is just pretty amazing. These are all the different cell types, and we're coloring in purple here by the neurons to give you a sense of just the immense complexity of neurons in the brain. And then to spatially localize all these individual cell types, Carol Balderrama serially sectioned 101 individual sections of a single mouse hemisphere to localize gene expression at 100 micron iterations across coronal sections of the brain. And that allowed us to then register all of these individual beads. So there are millions and millions of individual pixels here that capture gene expression. She could register those to the common coordinate framework and now identify which particular region each one of these beads was found. So the main heroics here computationally was to integrate these data sets together. We've got thousands of cell types and millions of beads. We have to position where all these cell types are across these individual beads. Jonah Langley led this effort, and he built a lot on the foundation developed by Dylan Cable, who actually I think will be joining you guys as an assistant professor next year. Dylan developed this great algorithm called RCTD for positioning single cell defined gene expression patterns in spatial transcriptomics data. We had to do a lot of adaptation actually, because just the scale of this problem is immense. To have thousands of individual cell types across such a large number of individual beads required a lot of engineering and some adaptation of the algorithm itself, but the overall principle was the same. And so to validate it, Jonah looked at the positions of excitatory cells in the cortex, which have this really nice striped pattern. And you can see now we're ordering these individual cell types by their spatial position along this perpendicular axis of cortex, because the cortical cells have this layer pattern. And so we can just look at their distribution. These ridge plots show their distributions, and we're ordering them by their known layer pattern based on data that was annotated by the Allen Institute. And so what you can see is that we're nicely recapitulating the expected positions because they're ordered in the expected way. We see this nice positioning across the layers of cortex, really demonstrating that we're nicely localizing cells with relatively minimal sort of false positive effects, which was an initial issue in the initial integration of these two data sets. So a sort of broad summary of these data are that the vast majority of the clusters we identified in the single nucleus data are neurons. And I should say the clustering was done in a very unbiased way. We just basically said, keep clustering, an iterative kind of clustering algorithm until there was a minimum number of differentially expressed genes defined for each individual cluster. And this is what the distribution looked like of each one of these individual cell clusters in sort of the major 12 areas of the brain. And you can see immediately there's this very intriguing thing here, which is that a very, very large number of these cell types and many, many small rare cell types are found in very relatively underexplored areas of the brain, like the medulla, the midbrain. These are brainstem areas. The cortex, which is very heavily studied by all of neuroscience, actually, we're quite oversampled here, and we're not seeing as nearly as much diversity as we're seeing in these other areas. This is just a more in-depth view where we're now looking at the thousands of cell types as rows of this heat map, and the columns are individual brain areas, and we're blocking them out by the major sort of top structure that are colored here in the bands on top. So we're marching from kind of anterior on the left all the way to posterior on the right. And you can see that most of these cell types localize to just one brain structure. And again, the vast majority of these cell types are found in these lower order areas, the diencephalon and the rhombencephalon, the interbrain and the midbrain and hindbrain. Zooming in an additional way, you can see that individual structures have just an astounding amount of variation. Here we're quantifying how many cell types are minimum needed to map to cover the 95% of all mapped beads. And so areas of the brain like the periaqueductal gray matter in the midbrain have hundreds of cell types inside of them. And we can validate this by looking at the individual cell types and seeing very discrete spatial localizations for many of them. Whereas the cortex are really topping out at 50 to 100. So there's a lot to learn about the brain because most of these areas are really not studied intensively by neuroscience. Almost all of the efforts in neuroscience are focused on these areas over here in what is called the telencephalon, the forebrain. So that's the main kind of message of the paper, and we did a lot of effort to kind of systematize these clusters, annotate them, provide minimum markers needed to distinguish them for generating tool lines for accessing them genetically. But I wanted to just mention some of the things that we've learned about the biology of the brain just from studying these individual profiles in the domains of understanding principles of neurotransmission, looking at activity-regulated gene expression. I'll explain what that is in a minute. And also utilizing this comprehensive atlas of cell types to start to look at where disease genes identified by studies like GWAS are preferentially enriched in different cell types. And this work was led by Nina Sachdev in the lab. So the first is a very simple analysis, which is just to look at neurotransmission in the brain. So the nice thing about the principles and properties of neurotransmission is that they require very specific genes. So in order to use glutamate, for example, as a neurotransmitter, you need one of these three genes, vGlut1 through 3. Or for example, to make GABA, you need a series of synthesis genes and a particular transporter. And so when we look at the coordinated expression of those minimally needed genes, we can identify particular cell populations as GABAergic, for example, versus glutamatergic. And what you see is that the vast majority of neurons use just one neurotransmitter, one of these fast neurotransmitters. There are some exotic ones in the sort of far tail of this upset plot that generate that utilize multiple neurotransmitters, but the vast majority are single neurotransmitter cell types. The picture is different and a little bit more surprising when we looked at neuropeptides. Neuropeptides are also genetically encoded, and they mostly have known receptors. So we could look at ligand receptor interactions as well amongst individual cell types. And the first thing we saw was that basically every single cell type, I mean, like 2/3 or 3/4 of all cell types or maybe more actually, more like 80 to 90%, I should say, are neuropeptidergic. That is, they make at least one neuropeptide gene. So this is a bit surprising because neuropeptides are thought to be, or at least often in the literature, thought to be more kind of specific to the hypothalamus. And even more kind of ubiquitous is neuropeptide sensing. All cell types express at least one neuropeptide receptor, and most express many more than that. So what's different about the different areas of the brain with regards to neuropeptide usage is the specificity of neuropeptides. So basically, there's a set of neuropeptides that are up here on the top that are highly specific to just a collection of brain areas where they were mostly discovered, like the hypothalamus, parts of the midbrain, and also in the brain stem, these more evolutionarily ancient areas. But then there's this collection of neuropeptides that are just ubiquitous, and they account for the majority of sort of neuropeptide usage in the brain. peptides like NPY, NTS, somatostatin. These are recycled and used ubiquitously throughout the nervous system. Most neuropeptide usage is between neurons, so neurons release and neurons sense based on our data. But there are some interesting examples of where we found examples of neuroglial interactions specifically through neuropeptide usage. So for example, angiotensin, which is a peptide that's used in the periphery to regulate blood pressure, it doesn't cross the blood-brain barrier, so it's made locally in the brain. And we detected it only in astrocytes, shown here in this dot plot. Its receptor is only found in neurons. So this is an example of where astrocyte neuron communication is probably occurring through this peptide signaling system. This is particularly interesting to us because we discovered in separate work that the angiotensin receptor AGTR1A is a very specific marker of the cells that are most vulnerable to Parkinson's disease in the dopaminergic midbrain cells. A flipped example of this is the peptide CART-PT, which has been studied a lot for its regulation in the context of activity and in substance abuse. And it turns out that this gene is produced, the peptide for this gene is produced quite ubiquitously across the brain, but maybe somewhat more enriched in the hypothalamus and the midbrain. But its receptor is very unique to microglia. So whatever this peptide is doing, and actually we don't know very much about the molecular actions of this peptide, it does seem like it's acting through microglia, which will be very interesting to explore in future work. So now I wanted to tell you about another analysis Nina did in a separate sort of biological exploration of the data, which is to look at activity-regulated gene expression. So when neurons fire heavily in the brain, they turn on a gene expression program called the immediate early genes. These are genes like FOS, the transcription factor FOS, as well as some of these other ones that are shown here. And the way that we harvested our tissue, the animals were immediately taken out of the cage and they were sacked, within 5 minutes. So the gene expression patterns that are present in the nucleus at the time of sampling really reflect whatever the animal was sort of thinking or doing at the time that it was removed from the cage. And so this gives us, and since we did nuclear RNA-seq, it was frozen immediately. So there's really no post-harvesting artifacts. So this gives us a unique opportunity to now explore de novo which genes are sort of co-regulated with FOS, which is the kind of canonical gene that we know is sort of ubiquitously activated by electrical activity in neurons. So what Nina did is she basically did a kind of a network analysis, a co-expression analysis, where she identified genes that were both highly correlated with FOS, but for which also FOS was a top correlating gene. So this kind of intersection of those two properties to form a network. And she identified a set of kind of core genes that are shown in red here, many of which are very famous in the sort of IEG molecular literature as being highly activity regulated, most particularly JUN-B. And then we could use this module as a way of just quantifying the relative expression of these IEGs across different brain areas. And we find that in particular that the telencephalic areas are more sort of FOS active at the time of sacrifice than these midbrain areas and hindbrain areas. Cortex being the most active of all when we look across the entire brain in our slide-seq data set. But now Nina wanted to ask whether there were particular cell type specific differences in how neurons respond to activity. So she took this network of correlated genes and she just clustered them based on their shared patterns of co-expression. And we identified a set of kind of core genes that were sort of ubiquitously correlated with FOS on the left here. And many of these genes that are labeled in colors are ones that had been discovered before as being activity regulated. I also point out this one in black here, which is a fun one, midolin, which was actually just discovered as an activity regulated gene in a science paper by Mike Greenberg and Steve Elledge's labs just a few weeks ago, actually. And so it turns out that it is involved in the degradation of FOS pathway protein partners. And this comes up as a very strongly correlated gene across the whole brain in our data set as well. But then we find some examples of genes that are much more specific either to only excitatory neurons in the brain, and these are in particular excitatory telencephalic neurons, because these colors here on the top reflect cells in the cortex and other related areas. And then a set of genes here that are more exclusively correlated with inhibitory neurons, or actually we see a module that's very specific to dopaminergic neurons. And it turns out actually that these inhibitory neuron specific genes are quite enriched for genes related to chromatin structure and modification. So we think that could be an important clue as to how inhibitory neurons respond to high levels of activity compared to excitatory neurons. A last analysis to share with you on this of this data set is a genome-wide association kind of localization analysis, in which we took the disease-associated genes that have been identified by a variety of GWAS studies and intersected them through an enrichment analysis with our single nucleus RNA-seq defined cell types, because we have now for the first time a direct kind of full comparative set of cell types, right? In these previous analyses that have done in this space, we haven't really known whether something is more enriched relative to the unknown, which is all the other cell types that haven't been sampled, or whether it is actually the most enriched in the brain. And so to do this, we leveraged a recently described single cell enrichment analysis, basically based on MAGMA, which was the previous sort of standard, but this is one that has been adapted particularly to single cell data that was developed by Alcus Price's lab. And Nina plotted these associations by cell type in a kind of GWAS-inspired way. So each one of these dots is a cell type in the brain, and on the left is the cortex, and we're going from the sort of anterior parts of the brain all the way to the posterior parts. The little yellow set of clusters there are the cerebellum, and then the gray cells on the right are glia. Since glia don't have the same regional localization, we just group them together on the right. And so for Alzheimer's disease, for example, you see nice enrichment of microglia and macrophages. This has been well described in many other analyses before and served as a nice reassuring positive control. And we see very different distributions of enrichments for different diseases. Bipolar disorder shows an enrichment in striatal and also in cortical excitatory populations. One other interesting one, autism spectrum disorder, shows some enrichment in the inferior colliculus, which is involved in auditory processing, which is often a challenge for autistic patients, and also in the bed nucleus, which is an area of the brain that is highly associated with social interactions and social memory. One particular one that we were particularly intrigued by is schizophrenia, which looks quite similar actually to bipolar. It makes sense because the two share a lot of genetic associations that were quite enriched for the principal cells of the striatum, as well as for the excitatory cells of the cortex. And the particular excitatory cells that are enriched in this case are ones that project to the striatum. So this particular analysis seems to suggest that this circuit of cortical striatal projections may be particularly relevant to the pathobiology of schizophrenia. So in summary, we've generated this whole brain atlas in which we've found that neurons of the brain show enormous diversification, but actually that this diversification is quite concentrated in relatively understudied areas of the brain, like the brain stem. And many of these areas, have only been cursorily studied from a molecular perspective. It's often thought that the brain stem is kind of a very static area of the brain that doesn't undergo any evolutionary diversification. But one thing to just point out is that in a recent study of ours last year, we profiled dopaminergic neurons from the mouse, rat, and intermediate with primates, the tree shrew, as well as in macaque and in humans. And we found that macaques and humans actually have a very unique cell type that's circled here that was not found in any of these other mammalian species. So this is a midbrain area. It's relatively evolutionarily ancient. It's in this part of the brain that is quite molecularly diverse based on our analysis. So we think that the point here is just that brainstem areas may not be interesting not only because they are diverse, but also because they may be continuing to undergo evolution even in relatively recent timescales. We also found that peptidergic signaling is a pretty ubiquitous neuron property, and we could use this data set in a comprehensive way to start to connect disease gene sets to specific cell type groups. Okay, So in the remaining time, I'd like to turn to a new technology that we've developed in the lab in close collaboration with Fei Chen's lab that we call slide tag. And I want to just set the scene for why we think this is so important. So single cell sequencing, as we all know, is this amazing suite of technologies for the following reasons. It's reproducible. It gives genome-wide data. It works in a variety of different tissue preparations. Many people have developed a fantastic set of computational tools to get a large amount of biological information out of these data. And because we're continuing to experimentally innovate on it to be able to do lots of different kinds of omic measurements. But the missing piece is spatial data, and that's the kind of motivation behind developing SlideSeq. And the main thing that it gives you, of course, is the spatial localization of cell types. And the current solution, in what I just showed you of the study that we did with the current approach, is to do both, and then to kind of munge them together with integrative analysis tools. But we would really like a technology that kind of gives us both, basically gives us single cell sequencing data with spatial information. And the question is, why is spatial data not sufficient? And I hope that the next slide gives you some sense of that. So here we're taking a set of data sets, both from our lab and from other published data sets of the brain, and we're just doing a UMAP analysis of cell types. And what you can hopefully appreciate is that the separation of cell types in the single nucleus RNA-seq data is far crisper. And you don't need to use UMAP to see this. You can do this in a very simple analysis of just taking the PCs of each of these individual data sets and looking at their relative eigenvalues, which gives you a sense of their relative information. And you can see that single nucleus RNA-seq relative to the others takes a much longer time to kind of peter out into this long tail than these other kinds of technologies do. And the reason for that is because, and this is also reflected, I should say, in marker analysis. If you just look at the markers are all a little bit mixed in these individual data sets. And this is the case both for imaging data, and it's also the case for sequencing data. The reason for the mixing is different. It has to do with segmentation versus pixelization, but the ultimate outcome for the analysis is the same. And that's the reason that we have to continue to rely on single cell data in addition to spatial data. And to tackle this problem, a set of great scientists, Andy Russell, Jackson Weir, and Naeem Nadaf, had the idea of taking SlideSeq arrays and kind of flipping them on their head. Instead of capturing RNA onto these beads that I'm showing here, barcoded beads, we took these barcoded beads and we photocleave off the barcodes and we deliver them into the tissue. These barcodes stick into the tissue and particularly into nuclei. And then we can harvest the nuclei and sequence the nuclei by regular single nucleus RNA-seq, for example, with the 10x Genomics platform. This is how the technology works in sort of greater molecular detail, but in essence, what you're doing is you're using the spatial barcode as a feature barcoding system in the 10x system, but we've also done this with just a standard poly-T capture of these barcodes and use that for in other platforms besides 10x Genomics. So this was the first iteration of this technology that really gave us a sense that it was working. We used the mouse hippocampus, which is just such a great tissue because it has this really fixed histological architecture. So this is what the data look like in sort of regular gene expression UMAP space, where you're just looking at relationships among cell types, and we've just colored them based on their annotations of the single nucleus RNA-seq data. But now each one of these individual cells has a collection of barcodes that it received from this photo cleavage of the array. And so it's not just one barcode that's informing a cell's location, it's a collection. And this is an example of one particular cell where a bunch of these barcodes are coming from this very, very small location here, and then a kind of noisy outer area here. And you can see the density of the individual UMIs of these barcodes shown. So this cell we can pinpoint as being located here by just simply taking this dense area and taking the centroid of where those particular spatial barcodes are coming from. And I'm just showing a little bit more detail on how this works. There's this kind of noise component here, which we get rid of by DB scan, where we basically just identify foci of barcodes, and then this kind of uniform noise in the background. We remove that uniform noise, and then we take the centroid of that cluster of barcodes, and that's our inferred spatial position. So this is what it looks like when we take that previous UMAP and we look at it in gene expression space. And the reassuring thing is that we see all of these same features that we expect to see in the hippocampus, the CA1 field, the CA3 field, and the dentate gyrus. We can even estimate the false localization rate because the dentate gyrus cells should not be anywhere outside of this strip, nor should the CA1 cells be anywhere outside of this strip. And based on that, we conclude that there's maybe a 1 to 2% mislocalization rate based on this analysis. So here are some key metrics that we've generated for slide tags to give you a sense of the data quality. It basically has indistinguishable representation of cell type proportions. Doing this, putting the tissue on the array, doesn't preferentially cause you to lose certain cell types. It doesn't seem to affect the overall capture of UMIs per cell. It's very, very similar. It doesn't seem to systematically sample one gene more than another. It's highly correlated between regular single nucleus RNA-seq and slide tag single nucleus RNA-seq. And the sampling density is on the order of 10% of all nuclei that we capture from a tissue section. So that corresponds in the postmortem brain, which we've been doing a lot of, about 7,000 nuclei that can be spatially placed in a 5.5 by 5.5 millimeter array. In terms of the actual spatial resolution of this technology, you can measure this in a couple different ways. One way is to look at a adjacent section with a histological stain like Nissl, and you can see this nice band of the CA1 field in the hippocampus. When we measure the thickness of that band by looking at a line that we fit to the center of it, and we compare that to the thickness of a line that we fit to the center of the slide tags data, we find that they are basically indistinguishable, which means that probably we're able to localize cells on the order of a single bead. But I will just say that we think that we're actually doing better than a single bead, because I told you that individual spatial barcodes, the individual cells, nuclei, are being positioned by a collection of barcodes. It's the centroid, the consensus centroid of many spatial barcodes, many pixels that contribute in that area. So kind of like GPS, you can kind of triangulate. And you can actually estimate the error just as you would with, let's say, super resolution microscopy by looking at the diffusion out from that centroid. And when we look at that diffusion, we find that most cells have a standard error, which is a measurement of the diffusion of around 3 microns in the X and Y axes. So this really suggests that even though the beads themselves are 10 microns, we're actually doing super resolution positioning using this array, which is kind of exciting and could be useful for many other applications as we get to kind of subcellular localizations in some rosy future. So in the remaining time, I'm just going to tell you about 2 applications of this slide tags technology that we're excited about. The first is sampling postmortem human cortex, and the second is an application to metastatic melanoma, which gives you a sense of kind of the multi-omic nature of this technology. So for the human tissue, one major challenge we've always had is the size of a human brain. When we did the slide seq on the mouse brain, we could kind of get away with a 5 or 6 millimeter sized array. But when you get up to human brains, it's actually obviously a lot larger. And so one nice thing about this technology is that it really scales nicely. And all you need to do is just kind of section a larger piece of tissue and put it down on a larger array, as long as you have the barcode complexity to be able to resolve all those individual nuclei. So in this case, we used a 1 centimeter by 1 centimeter array. And we took this piece of cortex, an adjacent section was histologically stained here, so you get a sense of it. This is the cortical layers. The inside here is where layer 1 is, and then on the outside is the white matter. That's the deep parts of cortex where all the axons go to other areas of the brain. From this initial analysis, we generated 26,000 spatially localized nuclei. You can see at the major cell class level, there are differences in spatial distributions. And the nice thing is that these data integrate very, very well with existing single cell data sets because they're the exact same data type. So you can just use regular Seurat to do the integration. You don't have to do anything fancy. And when you look at the individual label transferred nuclei from the slide tags data, you can see that if these individual cell types, we align this to an Allen Institute reference data set that has layer labels for every one of these excitatory cell types in the cortex. And what you can see is that when you look at their spatial distributions, they really, really nicely match what the Allen Institute annotated for these cell types, starting from the inner parts here, this is layer 2, 3, down to layer 3 and layer 4, 5, and then layer 5 and 6, and finally to the deepest layers of layer 6, you can see on the bottom here. So really nicely validating that the label transfer works well, that the cells are very easy to integrate with existing data. This also works for other cell types like inhibitory neurons and astrocytes that show different kinds of spatial patterns. We just summarized all that data in this ridge plot to show you again the spatial patterns and how they compare to the annotations from the actual Allen Institute data on the y-axis. So we were able to discover some interesting new laminar patterns. I talked about excitatory, inhibitory, and astrocytes, which are known to have layered patterns in cortex. But oligodendrocyte precursor cells actually have never been identified as having any spatial patterns. But when Matthew just ran an analysis where he looked for genes amongst the oligodendrocyte precursors, these are cells that make more oligodendrocytes in the cortex, he found that there were a set of genes that were more concentrated in the deeper layers and in the white matter compared to the gray matter and the upper layers. And so you can see that here for some of these individual genes, like this disease gene, GPNNB, which is very closely related and associated with Alzheimer's disease, and some of these other genes, which are more in the deeper layers of cortex, like SCV and SEMA3E. So I should say this is an example of a gene expression distinction that would never be found with just regular single cell, because for whatever reason, whenever you cluster the data, there's not enough of a variance here to really pull those two features apart. And so you can only do it by conditionally testing on whether a cell is in one particular layer versus another. So lastly, I was going to tell you about metastatic melanoma, and to showcase one other really desirable feature of slide tags, which is that it's not just restricted to RNA-seq measurements of cells. It can also be adapted to many other single-cell omic tools and kits that have been developed already. And in this case, we're leveraging the 10x multi-omic kit, which simultaneously measures RNA-seq and ATAC-seq from the same cells. So we used a resected, freshly frozen metastatic melanoma sample from a patient. This melanoma sample has a histological architecture that looks like the image shown here. This is an adjacent section. We first just did regular single nucleus RNA-seq on this just to get a sense of the cell types that were present. And Jackson did this analysis, and he identified a set of normal somatic cells that are on the left here, and then on the right in the dark blue and in the light blue are these tumor populations. We saw two very molecularly distinct populations that I'll come back to. We call them tumor 1 and tumor 2, and here's how they're spatially distributed. So the first thing you can immediately appreciate is that they're highly spatially segregated from each other. So there's this tumor population on the upper left, and on the bottom there is another tumor population that is highly separated in space. So Jackson was interested in understanding what are the differences in these two neighborhoods of cell types of tumors. So of the somatic cells that are found within and immune cells that are found within these two different tumor areas, what's different about them? And what he found was that if you just take the T cells in these two areas, you find that the T cells, the CD8 T cells in tumor 2 have a much more cytotoxic phenotype than those in tumor 1. And the cool thing is we don't just have to stop at these gene expression comparisons. We can now amplify the T cell receptors from these individual T cell populations from our library and look at both alpha and beta chains in our sequencing analysis and then connect them to individual cells. So now here we're identifying a particular clone with these protein sequences shown here. And we find that this particular clone is highly localized just to tumor 2. So we've been able to identify T cell clones with specific spatial localization using this technology, and then simultaneously also be able to profile their gene expression patterns within in situ within this particular tumor area. We next deployed the 10x multi-omic platform to look at epigenetic regulation within an adjacent tissue section. So this is the basic sort of summary statistics where we're able to see pretty decent TSS scores, a reasonable number of unique fragments per nucleus. giving us confidence that we're able to appreciate, broad epigenetic patterns from the ATAC data. Here we're looking at a correlation between differences in tumor 1 versus tumor 2 of log-fold change in gene expression patterns and also in chromatin accessibility, to give you a sense that these are correlated, and that we also identify sets of genes that are coordinately upregulated and open versus closed and downregulated in these two different tumor populations. And one of them particularly stuck out to us, this gene called TNC, which is highly expressed and sort of more epigenetic and more open in tumor 1 compared to tumor 2. And that's just shown here in space, where you can see these individual cells have very high accessibility. But the interesting thing to us, so first of all, we were interested in TNC because it's known to be connected to mesenchymal transitions in melanoma. And we also were intrigued by it because this pattern showed sort of this kind of clustered representation where only a subset of tumor 1 showed this epigenetic signature, whereas the other ones these gray ones here did not show this signature. And indeed, when we looked in UMAP space at a mesenchymal-like and a melanocytic-like gene expression score for these particular populations, we find that within tumor 1, which is where you see this kind of salt and pepper distribution of this TNC chromatin score, we find this kind of continuum in which some cells show more mesenchymal-like transmission, whereas others show more melanocytic-like transition. And we find that there are a set of transcription factors by looking at the particular enhancers that show this preferential accessibility in the mesenchymal like cells. We find that there are a set of transcription factors like FOS and JUN that are highly correlated. Their activity is highly correlated with this transition, which makes sense because FOS is a known regulator of mesenchymal transition in melanoma. So in summary, we've developed this platform for doing single nucleus spatial genomics. The resolution of this platform is on the order of 10 microns, but probably less based on this super resolution property that I described. We're able at this point to capture about 10% of nuclei in a 20 micron section. That's the standard section thickness that we do in a 10x genomics experiment. And the thing we really like about this technology is that it plugs into existing workflows. We're able to use single nucleus RNA-seq, a multi-omic platform, as well as others that are being developed now by utilizing the same barcode structure with our tag platform. I want to just emphasize what we see as some really key advantages here. Just like single cell, it's very easy to adopt. You don't need any microscope equipment or anything. You just need the array and the knowledge of where those barcodes are on the array. It has a seamless integration with existing single cell data and computational tools because it's basically the same data type. This is key. It segments the data for you because you're capturing individual nuclei. And I think this is a key feature of spatial transcriptomics data that is underappreciated. And is the main reason why we continue to need to do both, sample both data types in standard experiments right now. For us, we like it too because in imaging of brain tissue, there's a lot of human brain tissue, there's a lot of autofluorescence, which is very hard to overcome routinely. And the other nice thing is that it's because it's whole transcriptome data, you get access to human genetic variation. So you can actually see SNPs and QTLs and things like that that you can't see in probe-based detection. So some things that we're excited to do is to apply this to do whole tissue atlases, particularly of human sections, which have been relatively inaccessible to date because of their size and also the relative degradation of postmortem tissue. Technologically, there are a lot of really exciting applications and extensions of this work. One thing that we're really focused on is FFPE tissue, because I think the existing kind of progress that's been made in single cell FFPE can be directly leveraged with this platform to immediately allow single cell FFPE to be done in a spatially aware fashion as well. So with that, I'll just conclude and thank all those who really made this work possible. On the Mouse Brain Atlas side, I recognized already Jonah and Nina, who really led this work, but we're very much supported by the data generation folks, Carol, Chuck Vanderberg, and others. I should mention also that the mouse brain data is available on a portal, braincelldata.org. I seem to have missed that slide. I'm sorry about that. But all of the data can be downloaded, interacted with. You can visualize the distributions of all the cell types. You can plot genes. You can localize to specific structures. There's a lot of very useful data there for you to explore if you're a computationalist or a neuroscientist. And then on the slide tag side, again, this is a very close collaboration with Fei Chen's group. The folks who really made this possible were Andy, Jackson, and Naeem. And with that, I'd be happy to take any questions.
Wow, thank you so much. There's going to be applause, at least for me, you can hear. Quite a tour de force. I really, really appreciate it. Just incredible. And we can open it up to questions. Vanessa, do you have a way to see if people are asking questions that you can call on?
They actually should be able to unmute themselves and ask questions.
I had one general question in the field, Evan. A lot of times we have people that are identifying novel cells using single cell technology. They'll all of a sudden identify 20 novel cells that we didn't know existed. I always wonder, what are those criteria when we're trying to determine that we have a novel cell versus just a cell that has a different state? What are your thoughts?
It's a really great question. And, with the existing approaches of just doing single nucleus RNA-seq, you can't really do that. Ultimately, it has to be kind of done by kind of more functional analyses of and kind of perturbations. So that's one reason why I think, obviously model organism work continues to be very important in this space. So for example, we know that time of day has a pretty strong effect on gene expression. And if you tried to, we were very systematic about how we sampled these mice. And so we always took them at the same time of day and had the same light dark cycles and all that kind of stuff. But there are probably other features of, mouse emotion, behavior, cognition that are transitioning cells in and out of particular states. So they're going to need new molecular tools to be able to address that. And we're also going to need to be able to just utilize different perturbations, stresses, and things like that to see how cell states respond. I think there's sort of two things to think about, two answers in the immediate term. The first is that, RNA-seq is telling you that there's a certain amount of molecular distinction based on the number of DE genes or the strength of the separation. And what we have seen routinely is that cells beyond some degree of separation are, in our experience, I mean, of course, it's just empirical, are almost always not states. There's some kind of, type that's been fixed and by type, I mean fixed in development and not transitionable in the adult is basically the definition that we give. So that's kind of how we tried to set the bar in terms of the number of DE genes. But of course, we don't know if that's the case for all different cell types, for example.
Great, thank you. Anyone else have some questions?
We do have a couple of things in the chat. First of all, Megan said, great talk. I missed the very beginning. Are these results published? And if so, would someone be willing to share the citations?
Yeah, sure. So I'm sorry, both studies are in press. So they're both at bioRxiv, so you can look on bioRxiv. The studies are in press next month, will be out in Nature, the first one, and then the second one is in press and should be out in the next couple months. But you should be able to get almost all the information on the bioRxiv preprint. And also the data, as I said, I think somebody put the link up, and I'm sorry I didn't put that that slide in, but it's braincelldata.org is the actual portal where you can get all the atlas data.
And it looks like we do have a question on how logical and early do you consider the integration of spatial metabolomics data with these modalities?
Oh, that's an interesting question. I just saw a really great talk from Gel Lamano over in Switzerland, whose lab has developed a spatial atlas of lipidomics in the mouse brain. So I think it'd be very cool to try to integrate those. It's obviously very challenging because when we do the deconvolution of slide-seq pixels, we have some estimate of the proportion of different cell types. Typically it's one or two cell types that are dominating the gene expression pattern, but we don't necessarily know if that same proportion is going to be equivalent in the lipidomics data. And it may be very, very hard to tease apart which lipidomic signatures are from which particular cell type. But I think it's a very interesting idea, and certainly one that would be great to explore, especially in sort of focused disease context, because we know that, of course, certain, like, let's say, lipid patterns are associated with Parkinson's disease, and we know where to look in particular in that particular disease. So that could be a very interesting one to start with.
Yeah, that's great. I mean, these are the kind of things we're looking at with mass spec imaging now too, right? And trying to register that with single cell and spatial analysis transcriptomics. I think those are great concepts. I guess I wanted to ask one more question. And again, I think you kind of alluded to this with the thickness you needed for the tags. Your sectioning, you have a lot of cells that you've cut through, nuclei you've cut through. So is that why you think you're only getting about 10%? Are these intact nuclei that you're getting out of there? And would manipulating your thickness help?
Great question. So we know that we have not pushed the limit of the thickness. Part of the reason we don't do greater thickness is that then we have a lot of uncertainty in the z-axis. So then, the spatial localization becomes a little bit complicated. We're working on ways of trying to get the Z data, the Z information, but so far we haven't been able to crack that nut. The 10% we know derives from several places. So the first is just in the dissociation. So one thing that we've been actively working on, and we made actually some substantial progress in the last several months, is just improving the dissociation. Because I think one thing that's appreciated by anyone who's doing these single nucleus experiments is that you're never going to run out of nuclei. I mean, most experiments, you get way more tissue than you ever need. And so all of the nuclei protocols have not been optimized for yield. They've been optimized for quality. And we have to really revise our optimization approach because we just need to be able to get as many as possible. And so we've been doing a lot of that kind of work of just changing buffers and things like that to try to improve it. And we have made some substantial improvements. So we think about 70% are lost at that step. Then 10x loses about 50 to 60%, 50 to 60% in that pipeline. We're interested in other approaches that claim to have much lower loss, like the BD Rhapsody platform claims that it's 80% retention. So we're really looking into that avidly. And then the last step is in the spatial placement. So we lose about 40%. They're still single nucleus profiles and they're valuable, but they don't get spatially placed. And we actually understand that now a lot better just in the last few months. And we've been able to improve the spatial localization algorithm now to get it more up to 60 to 70%. And we may even be able to get higher. So, we've been able to improve it above that from the 60% retention now up to more like 80%.
Great, thank you so much. Any other questions? You can type in or shout out. Don't be bashful. All right, I guess not then at this stage. I think you wowed people so much. Incredible. Thank you very much. And with that, we'll sign off and really appreciate your talk. It was just incredible. And we'll be in touch again.
All right, take care. Thanks very much.
Thank you all.
So much,
Bye-bye.
Evan. It was a great talk. Thank you.
Bye.